And to close out our first session of talks, I'm Matthieu Nonnenmacher from Voyager.
I seem to keep being programmed to talk just after Ben year after year, and it never gets any easier, and that should be the story of my life, pretty much, that I always seem to come after Ben does his discoveries. And this is no exception. So we also have spent about the last 8 to 10 years at Voyager trying our best to develop efficacious and safe capsids for systemic gene delivery to the CNS. And this is the very rationale why we decided to create the TRACER platform. So there's 3 major routes today for delivery of genetic material to the CNS, and each one of them have pros and cons, and each one of them can be very appropriate for certain types of indication. And we have tried quite a few at Voyager before we actually decided to focus exclusively on in vivo delivery, on intravenous delivery, sorry. So one of the earliest ways for delivery was local. The advantages of those are the use of very low doses of virus, and also the lack of peripheral exposure, as well as presumably the capacity of the vector to bypass pre-existing antibodies that might exist in a fraction of the human population. The inconvenient, of course, being the invasive modality together with an exclusive zonal distribution. Second approach would be intrathecal or ICV, so CSF delivery. So this approach is kind of an intermediate one in terms of dose. Those are lower than intravenous capsids, at least for natural AAV capsids. The main inconvenience being that the distribution throughout the brain tissue is not fully homogeneous, and there's typically a gradient that is observed from the cortical regions, and that is pretty steep, and there's very little transduction in general to the deep brain regions. The advantages, of course, are low peripheral exposure and neutralizing antibody bypass. And then the last one, the intravenous delivery, which is arguably the most difficult, taking advantage of the very dense network of blood vessels in the brain to deliver in a very homogeneous fashion through every single area of the brain. Of course, the inconvenience are a higher dose, a potentially very high exposure of peripheral tissues to potential toxicities, as well as the sensitivity to neutralizing antibodies. And so these are aspects that we have tackled one after another at Voyager to try and progressively de-risk all these aspects for intravenous delivery. And so our goal was really to address all these particular challenges, achieve, of course, broad CNS distribution at well-tolerated doses, reducing the liver exposure, and also potentially evading partially or fully pre-existing antibodies. And I'm going to address every single one of these aspects in this presentation. So the TRACER platform was a very simple idea of implementing over existing screening techniques for AAV libraries, and it relies on library RNA recovery under the control of a neuron-specific promoters to really select the capsids with enhanced neural transduction through an iterative selection process directly in non-human primates. It was absolutely central for Voyager to obtain primate capsids, and as you will see later, capsids that allow also translation across species, including rodents. And so we were lucky enough to stumble on these first-generation capsids. The reason why I'm saying this is because it's exceedingly rare in AAV capsid development to get variants that work in both rodents and primates, and this capsid was one of those. It was displaying almost an identical brain transduction enhancement versus AAV9 in non-human primates and in mouse, about 50-fold. And the receptor for that capsid was identified as ALPL, which is highly expressed on the vasculature of the brain, as you can see on those stainings in human samples. And this gave us an incredibly high level of confidence for the potential of translatability of these capsids from rodents to non-human primates to humans. We were able to demonstrate very convincingly the binding of our capsids to the human isoform of ALPL, so direct physical binding, and also the functional interaction in a transduction assay that allowed us to see that the functional engagement of ALPL by our capsid was working with the cynomolgus isoform, the mouse isoform, and also the human isoform. And so we have very good reasons to believe that those capsids should perform in patients exactly the same way that they perform in mouse and non-human primates. Now, those capsids were still a bit limiting in terms of potency. 50 times higher than AAV9 is sometimes not enough. And so we performed an iterative evolution process towards the second generation capsid that allowed us to really establish a very strong therapeutic window, because at this capsid at a low dose, this was 4E12 viral genome per kilo in non-human primates, was capable of transducing more neurons in the brain than it did hepatocytes in the liver. And so this really started to look like a very good therapeutic window for gene transduction. You can see here that in monkeys, the gray matter of the brain was transduced very efficiently in almost all the regions that we analyzed. The capsids were capable of reaching about 40 to 60% of neurons and more than 80% of astrocytes throughout multiple brain regions at a dose of 313 viral genomes per kilo. The spinal cord showed more than 90% transduction of motor neurons, and the liver exposure was reduced by up to 30-fold compared to AAV9. So really extremely attractive properties for clinical development. And in addition to this, the lack of cell type bias, the versatility of the capsid really allows you to target whatever cell population you want by just switching the control elements in your viral genome. So this is the same capsid at the same dose with just genomes containing a ubiquitous promoter, a neuron-specific promoter, an astrocyte-specific promoter, or an oligodendrocyte-specific promoter. And what you can tell is, depending on your indication, your disease, and your target cell population, this offers a very nice blank canvas, let's say a very versatile tool. We put those capsids to the test for one of our leading gene therapy programs, clinical program for Alzheimer's disease that relies on down modulation of the tau RNA by a vectorized microRNA. So these are self-complementary genomes that express a microRNA against MAPT under control of a ubiquitous promoter in that particular case. And this is what happens to the tau RNA after 5 weeks or 11 weeks of treatment in non-human primates, following injection with a dose of 1.2013 viral genomes per kilo, if I remember correctly. And so the RNA went down by about 60% throughout multiple regions of the brain that are highly relevant for Alzheimer's disease. And the same was observed with the tau protein that was reduced by 50 to 60% following treatment with that capsid. So this worked extremely well in a real-life situation for a clinical program, and when we were very, very excited to pursue with that particular capsid. Now, as we painfully learned lately, there's obviously still some very real safety concerns about using AAVs in humans. And even though the primate models are far from perfect in terms of safety assessment, we still did our best by evaluating the elevation of liver enzymes as well as neurofilament light, so which are respective markers for liver tox and neurotox. And we were really happy to see that those capsids were extremely well tolerated, and we didn't observe any elevation of those toxic markers, at least beyond the range that is considered as perfectly safe. So this is not true for all the programs. There are some payloads that show some neurotoxicity, but at least in the tau program, we didn't observe any of those. Just to finish very quickly, so to go back on the theme of pre-existing neutralizing antibodies. So this is a true problem for AAV IV dosing. It is thought that about 30 to 50% of the human population harbors pre-existing antibodies due to childhood infection with natural AAVs. And we've been able to repurpose AI and machine learning, in our case, to engineer capsids that were accumulating the maximum number of mutations in potential epitopes without losing their fitness and their capacity to assemble. So this is the ML library that is here, including in regions that are structurally very important. And we were able to generate capsids that could evade human IVIG at a dose that completely neutralizes the parent capsid, the VCAP Gen 2. This was also validated in a pre-immune NHP with natural antibodies against AAV9, so that was also neutralized the Gen 2 transduction, but not the stealth capsid. And by analyzing human serum samples, we were able to see that those stealth capsids were able to evade roughly 50% more patient serum or human serum in general than the starting capsid. We have performed more cycles of mutation and epitope removal in these capsids, and now we think that we could more than double the eligible patient population with our new stealth capsids without any significant loss of brain transduction. And so this is something that is modular. Those modifications can be applied to muscle capsids, to brain capsids, because they are typically done in surface regions that are not important for the tropism. And so we are very excited at the idea of basically translating those modifications in multiple capsids that we are currently developing. So to make it short, we think that the TRACER platform has been quite successful in developing clinically relevant capsids that are de-risked as much as it is humanly possible. They work across species, they have an identified mechanism, and they show no sign of early or late toxicity following relatively high dose. We went up to 70-13 in non-human primates without any toxicity. And we are pursuing development of future capsids based on antibody evasion, as well as lower and lower doses. The ideal between being to hit sub 1E13 VG per kg capsids that could still maintain efficient targeting of the brain. So I think I'm already beyond time, sorry about that. I just wanted to thank the team that has been involved in all this work, and I'll stop here. Thank you.